everyone present here. Me and my group members are going to represent a documentary video about pollen fertility. Pollen, the tiny often invisible grain carried by wind, water or insects, plays one of the most vital role in the continuation of plant life on earth. Yet behind its delicate structure lies a complex world of fertility, genetics and survival. The documentary explores the fascinating journey of pollen from its formation within the anther to its union with the ovule, uncovering how environmental conditions, genetics and physiology determine its fertility. Join us as we delve into the microscopic world where life begins, where the strength and viability of pollen decide not only the success of reproduction but also the future of entire plant species and ecosystem.
Here we have the parts of flower. This beautiful flower consists of different structures: the petals, sepals, buds, stigma, style, ovary and stamens, each playing an important role in reproduction. Now let's look at the dissection of the flower. In the first image we can see bud and petals of flowers. In the second image we can clearly see corolla, corolla tube, style, stigma, stamens and sepals. In the third image, the sepal and corolla tubes are visible and highlighting the outer and inner floral layers. In the next image shows the ovary being separated carefully to observe the internal parts. Finally, in the last image, you can see the dissected parts: bud petals, stigma, style and anthers, all neatly arranged on the slide for observation. Let's begin. To study the pollen fertility of Catharanthus roseus, for this experiment we require the following materials. First, buds. Fresh flowered buds of Catharanthus roseus. Second, glycerol, which is used to prevent slide for drying. Third, microscope, to observe the pollen grains clearly. Fourth, cover slip, to gently cover the specimen on the slide. Fifth, acetocarmine stain, for staining the pollen grains to observe fertility. Sixth, slide, on which the specimen is prepared. And seventh, needle, for transferring and spreading the pollen on the slide. These materials are used together to prepare a slide and examine the pollen fertility under the microscope. Now, we have kept the buds of Catharanthus roseus dipped in water. Here, we have taken acetocarmine in one beaker and glycerol in another.
Pollen fertility in Catharanthus roseus is used to test the ability of pollen to fertilize the ovule and form seeds. Only healthy and viable pollen with an intact membrane can germinate and take up acetocarmine stain, turning deep red. Sterile or non-viable pollen has a damaged membrane, so it stains lightly or remains colorless. Acetocarmine is used because it helps to identify viable pollen easily. Bright red color indicates fertile pollen, while unstained or pale grain shown non-viable ones. Fertile pollen, while unstained or pale grain shown non-viable ones. As observed in this video, three different molecules, acetocarmine, DNA and phospholipids, interacting with each other. Acetocarmine binds to the both DNA and phospholipids because it carries a positive charge, which allows it to attach the negatively charged parts of this molecule.
Let's begin with the procedure. With the help of needle, we will remove the anthers of the Catharanthus roseus bud and place them over the glass slide. With the help of needle, we will squeeze out the pollen from the anthers. As we saw, there was a multiple anthers and stigma style placed over the glass slide. Here, we will put few drops of acetocarmine on the mass of pollen. Now, we'll wait for a minute and spread the pollen evenly over the slide while they are still deep in the stain. Now, we'll put few drops of glycerol. As we know, glycerol is slightly viscous, so it helps to mount pollen grain on slide, which make more stably and clearly for observing pollen shape, size and fertility. We'll cover the pollens with cover slip and observe under the microscope. Here are the results. As we observe the viable pollen and non-viable pollen, thus viable pollen appears bright red color. This is due to observed by the cytoplasm of living pollen grain. And non-viable pollen, the cell cytoplasm has degraded, so the pollen grain stains poorly. Thank you everyone for your time.